nervous system. In this lecture, we're going to start by talking about the overall purpose of the nervous system and go through a general, very general anatomical overview. We'll then go over several kinds of cells involved in the nervous system, that being glia and neurons. We'll talk a little bit about the different kinds of glia, not very much. And we'll then start going over how membrane potential is relevant to the nervous system. We'll start with the idea of resting membrane potential, and then talk about two different kinds of changes in membrane potential, which you can think of as nerve signals, that being graded potentials and action potentials. Let's talk nervous system. Now, I have to admit, despite the fact that I am quite enamored of all of human physiology, and I love teaching this stuff. The whole body is fascinating, and every part of it is really neat to learn about. This is kind of my favorite. Because one of my main interests personally is the notion of the brain and how it produces the mind, and how our minds exist within a body. And as far as we can tell, the mind is a function of the brain, which is part of the nervous system. So this is an area that I am particularly interested in learning about. And I love teaching about it. So let's start by talking about what is it that the nervous system does? Why do we have it? What's it for? What purpose does it serve in our body? Now, one of the main things is that we often need to get signals to coordinate functioning of the body. A lot of that coordination comes through the nervous system. So, coordinating body function. Now, that coordination means bringing in information about what the body is doing, as well as information from outside the body. So, in a way, this actually touches on one of the more interesting bits of philosophy about the nervous system. We're going to consider the fact that it's our brain and spinal cord, what we call the central nervous system, we're going to get to that makes the decisions about what to do in our body. I guess subtleties there. We'll talk about the enteric nervous system a little later. But in general, the pattern is sensory information, either from within the body or from outside, comes to the brain and spinal cord. Some sort of processing is done, and then a response comes out from there. Now, for some things, like reflexes and reaction, like reflexes, which we will talk about later, there's not really a lot of decision-making. Signal comes in and it produces an immediate response, kind of at a hardwired level. But for some things, for reactions, there's some level of processing that has to be done. We have to take in that information and then analyze it, decide what that information means, recognize the pattern of that information, and figure out what the appropriate response to do would be. And so part of this is bringing in sensory information. So, for example, what I'm doing right now and what you're doing when you're looking at this, if you're looking at this, is there are patterns of light and dark coming to your eyes. Those get turned into nerve signals. That process of taking sensory information and turning it into patterns of nerve signals is called sensory transduction. That's a job of the nervous system, to take information and turn it into patterns of nerve signals. And then to take those nerve signals, transmit them to the central nervous system, and then the central nervous system has to recognize patterns. It has to process that information and figure out what it means. So that's a matter of processing and pattern recognition. And then we have to integrate that into our model of the world. So somehow my brain has to have some internal model of where I am and where the world is and what the world is doing with me and how I work within it. So when I close my eyes, I know that there's a whiteboard a few inches behind my head, and if I move my hand to here, I'm probably not going to touch it, but if I move it much further, there it is. And I have a sense of where the door to this room is and all of that. I have this model in my head of where my body is and where the world is. And part of the job of my sensory information is to update that model, to make sure that my model is accurate so that it can guide my responses. So I have to maintain a model of the world and of myself. I have to have some model in my head of myself, both my body and even, and this is kind of interesting, my own mind. I have to have some level of self-awareness of the fact that I am thinking about things. It's kind of there's some interesting philosophy in that one. But anyway, and then, once I've decided how to respond to that sensory information, I can issue commands to the body. So, all of this is part of what our nervous system is for. To take in information, to process it, to determine the best response, and then to coordinate those responses, to send commands to muscles and glands, to get the body to do things in response. A lot of that is about maintaining homeostasis. And not all of this is conscious. In fact, most of it isn't. My nervous system has to be responsible for taking in information about what's in my digestive tract and where it is and what kind of responses would be best to do. Also, some of these responses are coordinated by the endocrine system, and we'll get to that later in the course. But right now we're going to talk about the nervous system. So that's kind of our overall picture. This is about getting information, bringing it in, recognizing patterns, determining responses, and issuing commands. To understand any part of how that works, there's several things we have to get through. We need to understand what cells are involved here, what kind of signals are involved, and how those signals are transmitted, and how they're received. So, to understand how the nervous system is going to work, we need to understand what it's made of, the cells that make it up, how they signal each other, what those signaling mechanisms are, how the signals are transmitted, and how they are received. Once we get that, we can start to understand a little more about how this system works at a mechanical level. So let's start by putting together an overall picture of the nervous system. So let's erase this. Now we'll start with kind of a very broad overview. We've got a brain, all the wrinkly bits, and a spinal cord. This is the central nervous system. This is where decisions are made. Most of them. So this is where the stuff comes in and where the decisions are made. Then, in addition to that, we have lots of things coming out from that. This is not meant to be anatomically accurate. We have lots of parts of the nervous system that are bringing information from the body and the world to the central nervous system. And also parts of that which are taking information out from the central nervous system out to the body. All of this stuff out here that's not the brain and spinal cord is the peripheral nervous system. Collections of nerve cells and parts of nerve cells that are either bringing information from the body and the world to the central nervous system or taking commands from the central nervous system out to the body. That's kind of the big division. And we're going to talk about subdivisions here, like the division of peripheral into somatic and autonomic and such like that. But we'll get to that later. Now, all of this is made up of a variety of types of cells. So, let's erase that. And talk about the different kinds of cells we have in this system. So, the two main classifications of cells in the nervous system are the neurons, which is what you probably usually think of when you're thinking of a nerve cell, and glia, which are cells which provide support for the nervous system. That can be structural support, making up physical structure, metabolic support, making sure that the neurons have the nutrients they need and don't get overwhelmed by wastes. It can be maintaining the cerebrospinal fluid, the extracellular fluid around the nerve cells. It can be providing immune-like functions in terms of patrolling the nervous system. All of those things are, it can actually also be altering the neurons so that they can transmit their signals more effectively. All of those are functions of glial cells. The function of the nerve cells is to bring information in, process it, and transmit signals. The glia are there to help make sure they can do that. So, let's take a moment to start with the glia. I'm just going to mention some types of glial cells and some of the things that they do. So, for example, astroglia. These provide structural and metabolic support for nerve cells. They also maintain the blood-brain barrier, which is both a kind of physical and a conceptual barrier between the bloodstream and the nervous system, where we exercise extra vigilance about what sorts of things can go from the blood to the brain. In most of the body, we allow some kinds of molecules to move fairly freely between blood and extracellular fluid. But in the brain and spinal cord, in the central nervous system, the astroglia, as well as some changes in the capillaries, provide an extra layer of vigilance about that. Only certain molecules are allowed to go from the blood to the brain. It's highly controlled. That's the idea of the blood-brain barrier. It's actually one of the reasons why it can be difficult to deliver pharmaceuticals to nerve tissue, because the blood-brain barrier doesn't let a lot of things through, even things which we would want to get through because they would help us. A lot of chemotherapy drugs, for example, don't easily pass through the blood-brain barrier. So, some interesting stuff there. Okay, other ones. Oligodendrocytes, great name. So oligodendrocytes and a similar thing in the peripheral nervous system called Schwann cells. One of their main functions is myelination, which is something you'll learn about when we talk about the propagation of action potentials. It's a modification to what we call axons, which are part of nerve cells, that speeds up how quickly signals can be transmitted. Also, we've got microglia. Microglia function a lot like immune cells. These microglia are involved in monitoring the nerve tissue, removing contaminants and invading microorganisms. They provide a lot of that same kind of almost like macrophages, although they're not, but they patrol the tissue and clean things up. And then there's also ependymal cells. Which are involved in maintaining the cerebrospinal fluid. Which is just the brain and spinal cord equivalent of extracellular fluid. It's the fluid around the neurons and the glia. The ependymal cells are involved in processing that fluid as blood plasma enters the brain and maintaining it. So, now how many glia there are, that's an area where there has been some dispute. The statistic I learned in grad school is that there are roughly 10 times as many glia as neurons. So for every neuron, there's about 10 glia of various sorts, which might be responsible for this notion that you only use 10% of your brain, which, just so you know, is completely false. You do not have 90% of your brain sitting there going, gosh, I hope this person develops psychic powers soon because I'm getting bored. You use all of your brain, pretty much all the time. Whether you use it as efficiently as possible and as effectively as possible, that's a different question, but you don't only use 10% of your brain. Only a portion of your brain cells are neurons, but it's not like the other ones aren't doing anything. These glia work very hard. Now, what the actual proportion is, not that important for us right now. There's probably more glia than neurons. It may not be 10 times. Doesn't really matter right now. All right, so now that we've talked about glia a little bit, let's talk about neurons. And there's lots of different kinds of neurons. The one you've probably seen most often, the drawing you'll see in a lot of textbooks, looks something like this. Looks familiar, maybe? This is one common kind of neuron. This is known as a multipolar neuron. Let's make sure we know some terminology for this, and then I'll show you just some sketches of a few different kinds of neurons. So, in a typical multipolar neuron, we have one area here. Let me change this just a little bit, make it look prettier for me. Okay. We have one area here that contains the nucleus and most of the organelles of the cell. The name for this thing over here might be cell body or soma. Either of those terms is acceptable. This is where you find the nucleus and most of the organelles, things like mitochondria. Usually coming off the cell body are small thread-like branching things that are involved in collecting information from other cells, or sometimes from other structures. These, collectively, are generally known as dendrites. Generally, the job of dendrites is to bring information in. We'll talk about how that works later, and dendrites have some interesting subtleties to them but we're probably not going to get into most of that. Most neurons have one long extension coming off the cell body that's involved in transmitting signals from the neuron elsewhere. The term for that is the axon. And then the axon will end at, this is showing 2, there's usually actually a little more than that, as in, up to several thousand, of these ending points where information will be transmitted from this cell to some other cell. There's a lot of different names for these. You might see them called synaptic terminals, or axon terminals, or terminal buttons, or terminal boutons. I've seen people try to keep it French. I usually go with axon terminals, but I'm probably okay with most of those. So, now one other one, the one that you might not be familiar with, although many of you probably are. The very first part of the axon, where a nerve signal called an action potential usually starts, kind of has its own name. We call this the axon hillock. So if you know those terms, then you probably know the basic neuroanatomy you need for this. We're going to get into what goes on at these different places shortly. Now, like I mentioned, that's one kind of neuron, what we call a multipolar neuron. It's a common type, but there are many, many others. And I'm not going to hold you responsible for naming them. That's more of an anatomy thing. I did want to show you some of them. So there are some. This is a pyramidal neuron. It has several groups of dendrites and then an axon that comes off of it. You find a lot of these in your cerebral cortex. This is a pseudo-unipolar neuron. You find this is a common type for body senses. Your touch sensation uses this type. That's a bipolar neuron. You find some of these, for example, in your retina, as well as some other places. Well, I'm not going to skip. There are many others. And in some cases, it's clear that you have dendrite, cell body, and axon, but in other cases, which of those is an axon? Which of those? I mean, in some cases, things get a little more confusing, but that's why we say the general pattern is what I showed you, but there are variations and subtleties on it. We'll even touch on a few of those as we go. So, that's kind of the general idea of the functioning of the nervous system and some of the cells involved. Now, the next thing we're going to need to talk about is the idea of how membrane potential is relevant to these nerve cells. And the first thing, the thing I want to do before we get into the details is give you a little bit of terminology. So, at this point, you already know something about membrane potential. In the previous lectures, we talked about what it was and the idea that allowing ions to cross a membrane can change the membrane potential. So membrane potential in general is the difference in charge between the inside and the outside of a cell. And if I let ions cross the membrane, that changes that difference in charge, which means it changes the membrane potential. We talked a little about how membrane potential is usually measured in millivolts, how for every ion that can cross a membrane, you can figure out what its equilibrium potential is, and then you can determine when you open a channel for an ion, the membrane potential tends to move toward that ion's equilibrium potential. We talked a little bit even about what happens when more than one ion can cross the membrane, how the membrane potential we get will be somewhere between those two ions' equilibrium potentials. So I wanted to, since membrane potential is so critical for nerve cells, it's how their signaling works, I wanted to give you a little bit more information about what we're going to talk about with that. So first of all, we're going to talk about the idea of resting membrane potential. That is the membrane potential that a nerve cell tends to go to when it's not actively doing some other kind of signal. Sorry. It's the membrane potential the neuron goes to when not doing anything else. You could think of it as sort of the base state of the neuron. Do keep in mind, as you'll see, when we say resting membrane potential, that does not mean the cell is exactly at rest. This is not a stable equilibrium. It's a steady state that the cell has to maintain, but like we talked about at the very beginning of the class, that's not the same as equilibrium. At resting membrane potential, you're like a balanced teeter-totter. You can cause the cell to do something just by opening or closing a few ion channels. It's just the state you tend to go to when you're not doing something else. There are then two kinds of nerve signals we're going to talk about. The idea of a graded potential and the idea of an action potential. And I also need to give you a little bit of terminology. So if we imagine a chart here with membrane potential on one side, this would be time. And let's imagine that this here is our resting membrane potential. If the cell's membrane potential becomes more positive than resting membrane potential, we say that that is what we call depolarization. Membrane potentials going more positive than resting membrane potential we call depolarizing. Membrane potentials going more negative than rest we call hyperpolarizing. These terms are not used entirely consistently. So for example, sometimes if something depolarizes and then goes back toward rest, we might say it is then hyperpolarizing. I've seen that term used, which gets a little confusing. I think it's a little better to say this is depolarizing, and then if it returns to rest, we might say it is repolarizing. So this would be repolarization. Now, after hyperpolarizing, what do we call that? I haven't actually heard a consistent term for it. We'll say going back from being hyperpolarized, maybe. In any case, there's some terminology you need to know. So a membrane potential change that is going positive, we generally call depolarizing. Negative, we call hyperpolarizing. Okay. That should about do it for the stuff you need for the basics. So in the next part of this lecture, we're going to start talking about these things: resting membrane potential and graded and action potentials.